This is part one of the lecture notes on cell communication. Let me set up the slideshow. I wanted to talk to you just a little bit about the image on at the beginning of the slides. What you're looking at is an image taken by recorded by a microscope, a scanning electron microscope, and it's interesting where these cells came from. They're actually human cells. They're cells from a female. They're cervical cells, and the cervix is the opening to between the vagina and the uterus. So these are actually cancerous cervical cells. And here you can see two that have just finished dividing. But this is, again, this image, you can't see this much detail with a light microscope, but this image was taken with a scanning electron microscope. Um, so as far as cell communication, there's two types. There's intercellular signaling, and inter, I-N-T-E-R, means between. So that means signaling between two different cells. And then there is intracellular signaling. That means signaling within the cell. Chemical signals are released by signaling cells, and usually they are called ligands. So the chemical signal is called a ligand, and then the ligand will bind to a specific receptor on the target cell, and some type of response will occur. So it's important to understand that a specific ligand will have a specific receptor that only binds to that ligand. Here are four forms of chemical signaling. There's autocrine, which means the cell signals itself. So the target cell is actually the signaling cell. There's also gap junctions. That is when a cell targets a cell that it's connected to by connections called gap junctions. And these gap junctions connect the two cells to each other, but they allow for certain molecules, normally certain types of ions, to pass through. Um, and then there's paracrine. So there's signaling across gap junctions. There is paracrine signaling, which is when a cell targets a nearby cell. This is very, very similar to endocrine signaling, which is when the signaling cell sends the ligand through the bloodstream to the target cell. So here, where the ligand is actually a hormone. Okay, so paracrine signaling, and what you're looking at is each of these cells are neurons, and if you read over on the left, signals that act locally between cells that are close together. That's the key with paracrine signals, is that the cells are close together. Paracrine signals move by diffusion through the extracellular matrix. These types of signals usually elicit quick responses that last only a short amount of time. So Here you have what's called the presynaptic cell, and the place where it meets up with the target cell. They don't actually physically connect with one another, but they get very close to each other. This is called the synapse. And what you see are vesicles in the presynaptic cell, the signaling cell, that contain neurotransmitter. Neurotransmitter is some type of chemical ligand. There's many different types. There's acetylcholine, there's norepinephrine, there's serotonin, dopamine, um, GABA. There are just many different types. But basically, the neurotransmitter is the ligand, and it is released into the space between the two cells and attaches to the receptor, which is right here on the postsynaptic cell. That postsynaptic cell can be another neuron, so one neuron sending a signal to another neuron, or it can be even a muscle cell, the neuron sending a message to a muscle cell to contract. So paracrine signals are when the two cells, the signaling cell and the target cell, are close together. Endocrine is very similar to paracrine in how it works, except that endocrine signals originate from endocrine cells, typically endocrine glands, and the ligands are specifically called hormones. In the nervous system, the ligands are called neurotransmitters, but in the endocrine system, the ligands are called hormones. And the most important thing is that the target is far away. So the hormones have to travel through the bloodstream some distance away from where they're released. Now, autocrine signals are when a signaling cell sends a signal to itself. That means the signaling cell and the target cell are the same, or else they are very similar. This type of signaling often occurs during the early development of an organism to ensure that cells develop into the correct tissues and take on the proper function. Another thing that autocrine signaling does, it regulates pain sensation and inflammatory responses. Inflammatory responses um, happen due to the release of a chemical called histamine, and that causes the 4 cardinal signs of inflammation. You may know these: heat, redness, pain, and swelling at the area of inflammation. And if a cell is infected with a virus, that cell can signal itself to undergo apoptosis or programmed cell death. So the virus won't spread. Direct signaling through gap junctions is a type of intracellular signaling. Um, let's see. And what it does, it allows a group of cells to coordinate their response. For example, cilia that line our respiratory tract, like say this is the cells of our respiratory tract. Here's a nucleus, and they're lined with cilia. Well, all that cilia is going to sweep up toward the opening of our respiratory tract, which is our mouth and our nose, so that the mucus that's in our respiratory tract has trapped pathogens and debris and germs, and it sweeps all of that harmful stuff up and out of our respiratory system. So those cilia are coordinated and move together, move the same way at the same time. So anytime you have coordinated movement, that is typically gap junctions that are coordinating that movement. There are small hydrophobic ligands, such as ligands that are derived from a molecule of cholesterol. Cholesterol can be recognized because it has one, two, three, four rings in its structure. Just a very simple change to cholesterol can convert it to testosterone and estradiol or estrogen, which you may recognize as the male and female sex hormones. Um, so these are steroid hormones. They can, because they're steroid hormones and they're lipids, they can diffuse directly through the plasma membrane of cells. Remember, if they are hydrophobic, that means they are lipid soluble and they can diffuse directly through the phospholipid bilayer. They don't need to move into the cell through a channel or carrier protein. Now, if you look at the second paragraph, don't let it confuse you. It does say hydrophobic ligands must bind to carrier proteins while they are being transported through the bloodstream. But as far as crossing the plasma membrane of a cell, they can actually cross directly through the phospholipid bilayer, and they will bind to a receptor inside the cell. There are water-soluble ligands, and if they're water-soluble, they are polar, and they cannot pass through the phospholipid bilayer of the plasma membrane without a carrier or channel protein or without coming in by a vesicle like exocytosis. Most water-soluble ligands bind to the extracellular domain of cell surface receptors. So most of the time they bind to a receptor on the surface of the cell. This group of ligands is quite diverse and includes small molecules, peptides, and proteins. Signaling receptors are protein molecules in the target cell on its surface that bind to the ligand. There are two types. There are internal receptors and there are cell surface receptors. Now what you're seeing here is an internal receptor, and you can see that the signaling molecule is hydrophobic. It is diffusing directly through the phospholipid bilayer and binding to that intracellular receptor. And then the intracellular receptor is delivering it to the DNA where it will cause a gene to be activated. So many intracellular receptors are transcription factors that interact with DNA and regulate gene expression. Cell surface receptors are just like it sounds. They are located on the surface of a cell, and they are involved in intercellular communication. So a ligand would bind to this cell surface receptor, and a cell surface receptor is always a protein that spans the entire plasma membrane. In other words, if this is your plasma membrane, it's a transmembrane protein. So your cell surface receptor might look something like this. And then your ligand might look something like this. And that's where it would bind into the cell surface receptor. What kind of signaling molecules bind to cell surface receptors? Well, it's ligands. ligands